Hey everyone, welcome back to Windows Seed Studies. I hope you all are doing well. So, we were discussing about cell biology, in which we have already learned about cytoskeleton, and then we started discussing about plasma membrane. So, by now, you already know that plasma membrane is a two-dimensional fluid. So, the model which is now accepted for the structure of plasma membrane is the Singer-Nicolson model, which is also termed as fluid mosaic model. So we know that why it is considered as fluid, why the plasma membrane is fluidic, because it is made up of the lipid bilayer, which is not immobile, which is actually a mobile entity, just like any fluid, just like liquid or gas, which moves constantly, which flows constantly. Lipid bilayer is also dynamic, and that is why the plasma membrane is fluid. But we haven't discussed about why this model is also known as fluid mosaic model. So, well, for that, let's have to go back in the history and let's learn about how the different researchers, when they were studying about plasma membrane, they got to know the mosaic nature of plasma membrane. So, so today we are going to learn about lipid rafts. Okay, and why the membrane is termed as fluid mosaic model. So why this model is accepted. So this lipid raft, this term, it was introduced in late 1990s. It was introduced when researchers they made their observation when they were studying the plasma membrane, and they solubilized the plasma membrane in non-ionic detergent. So we already know that the plasma membrane is made up of lipids. So it should be solubilized in the detergents. So when researchers used non-ionic detergents, there were certain parts, or let's call them microdomains, which did not solubilize in those detergents. So there were certain parts of the membrane which resisted the solubilization, and that the parts that resisted solubilization, they were termed as lipid rafts. So the lipid rafts are the microdomains in the plasma membrane or of the plasma membrane, which are detergent resistant. Means they did not do, they does not get solubilized by the usage of detergent on those plasma membranes. So those are, they are also known as DRM, detergent resistant membranes. So the fact that the such microdomains exist in the lipid bilayer, that is why the lipid bilayer, the plasma membrane, is also known as fluid mosaic. So what is mosaic? Mosaic is when the small parts, small bits of tiles or small bits of papers, they come together to form a pretty picture. So I have a picture of that. So here you can see when different colored small pieces of tiles, they have come together to form this pretty picture. So in this picture, even though this is one picture, one single entity, but this is compartmentalized in different micro domains. So you have this different tile, this different tile. So this is a mosaic. So many bits of some one entity, so many bits of pieces, like many pieces of tiles here, tiles or many pieces of papers, they come together, which have certain characteristic property. For example, this tile has blue color, this tile has green color. So each microdomain has its own distinct property, and those different types of microdomains, they are coming together and they are forming one entity. So let's imagine this in the context of lipid bilayer. So lipid bilayer is one entity, and that has different types of microdomains which have different characteristics. Means in this case, by characteristics I mean the different composition. So that composition, it is not fixed, that is variable, but the characteristics that arises from that composition, that is same, that the lipid rafts will be detergent resistant. So one characteristic is same, but the composition in that lipid raft, that will vary. right? So those compartmentalization of lipid bilayer, due to which it looks like a mosaic, multiple pieces of what microdomains, they are coming together to form lipid bilayer. It looks like that, and that is why this lipid bilayer, that is also termed as fluid mosaic. So when researchers, they were studying about the liposomes, they were studying the organization of plasma membrane. When they used, they used, so we already know that plasma membrane is made up of different types of phospholipids, sphingolipids, and sterols. So they used different types of compositions of those lipids, and they saw that what kind of membrane is formed. So they were forming liposomes using different compositions. So when they used sphingolipids, as well as the the phospholipids here, let's say if they used phosphatidylcholine. So when the ratio of them was kept 1:1, so when they only used those two lipid bilayers, the liposomes they were formed, they were uniform. Okay, they were uniform in structure, just like any kind of solid ball in imagine a 3D solid ball. So it looked like that. Under microscope, we know that the lipid bilayer is fluid, but it looks like solid. But when you mark certain points, so let's say like I mark this certain point in this solid ball, which looks like a solid, but when after certain time t, it will float and it will go from here to here. This means it wasn't a solid form membrane, but it was actually fluid. So this is how the sphingolipid and phosphatidylcholine, when they were mixed together in equal volumes, they formed a uniform bilayer. But when researchers added sphingolipid, phosphatidylcholine, as well as cholesterol in the mixture, so all of them were kept 1:1 in the ratio. So the composition, they had equal proportion in the liposome formation. Then the liposome which was formed, it was actually mosaic liposome. So I also have a picture for this. Here you can see. So this is first case where the sphingolipids as well as cholesterol, the phosphatidylcholine, they have 1:1 ratio. But here, they also added cholesterol. So sphingolipid, cholesterol, and phosphatidylcholine, all of them, they are present in equal proportion. So the membrane that was formed, the liposome that was formed, it was mosaic. So here you can see clear microdomains. Okay, so darkly stained are the microdomains. Even those, lightly stained doesn't mean that this is empty space. Membrane is present there, but it has lower thickness compared to those darkly stained microdomains. So this is how the structure looked like. So liposomes were, they were mosaic. While when they did not add cholesterol, the liposomes did not, they formed a uniform structure. This is because, as we discussed in the class where we were discussing about different types of lipids that are present in plasma membrane, we discussed that cholesterol, which has a steroid ring, it is actually also considered as a membrane fluidity buffer because it has dual properties. It can either increase or decrease fluidity depending on the conditions, depending on the environmental conditions. But basically, cholesterol improves the rigidity. So due to having due to having a sterol ring, cholesterol has a unique property to rearrange certain lipids together. So it rearranges those sphingolipids, and those saturated lipids together in certain domain, which results in the formation of such microdomains. So this is a distinct property of cholesterol, and due to which those microdomains are formed, and those microdomains, they were termed as lipid rafts. So this term dates back to 1990s. So in the late 1990s, this term was coined. But well, there are a lot of controversies about the terming lipid rafts. I'll discuss that again in a while. So basically, in short, the microdomains that are present in the plasma membrane, which are detergent resistant, means the membrane lipids and proteins, they do not get solubilized in the detergent, in the non-ionic detergent at low temperature. Okay, at high temperature they can get solubilized, but at low temperatures, and this experiment is done at low temperature, and they do not get solubilized. Those microdomains, they are referred as lipid rafts. Okay, and due to the presence of lipid rafts, cell membranes, they are not homogeneous like we previously saw. They are actually fluid mosaics which are laterally compartmentalized. Okay, so I hope you get the concept of why we call lipid bilayer as fluid mosaic. So let's now learn about the molecular composition of lipid rafts. What is the molecular composition? Okay. So the lipid rafts, the core components of lipid rafts are cholesterol, okay, then sphingolipids, majorly sphingomyelin, okay, and saturated phospholipids. So I hope you have seen the previous lecture where we discussed about the components of plasma membrane. What is the composition of plasma membrane? If you have seen that video, you will be able to guess why only those components, those different lipids are the ones which are present in higher concentration in lipid rafts. Because cholesterol adds rigidity in membrane, and that is why it is going to improve the or it is going to decrease the fluidity in that certain part. So it is going to add the stability to lipid rafts. It is going to make it rigid, and that is why it makes them difficult for getting solubilized in detergent. Then sphingolipids such as sphingomyelins or the glycosphingolipids, which have long saturated fatty acid chains, they are again going to promote tight packing, just like saturated fatty phospholipids. So phospholipids, on the contrary, unlike the unsaturated phospholipids, which have a kink in them, saturated phospholipids, they promote the tight packing. So all of those different lipids, all of those, they are going to promote the tight packing and they are going to enhance the rigidity, and that is why they are present in lipid rafts. Okay, so they are going to enhance membrane rigidity, and that is why they are present in higher concentration in lipid rafts. So alongside the proteins that are present in the lipid rafts, there are alongside those lipids, there are proteins that are associated in lipid rafts, such as GPI-anchored proteins, transmembrane proteins, and palmitoylated proteins, the proteins which have palmitic acid as their anchor. So I'll just list those proteins here. We haven't discussed about membrane proteins yet. So when in next lecture we'll discuss about it, you'll get to know why only those proteins are present. So let me just list them down here. So GPI anchored proteins are there in lipid rafts. Then there are certain transmembrane proteins, and there are palmitoylated proteins. Okay, so those are the ones which are present in the lipid rafts. So alongside those, there are certain signaling molecules which are also present in the lipid rafts, and they are very important because due to them, the downstream signaling is going to start. So there are SRC family kinases, then there are caveolins, flotilins, okay, flotilins. So those are the signaling molecules that are present in lipid rafts. So as I say, that lipid rafts, they do have certain composition, but this is not fixed. They will have cholesterol, sphingolipids, saturated phospholipids, but the composition, like like I said in the liposomes, it was 1:1:1. This composition is not quite fixed in lipid rafts because rafts are defined more by their lipid-lipid or lipid-protein interaction than by fixed composition. And that is why this is a topic of controversy. And it is now redefined. The lipid rafts, they are now redefined as the dynamic, the dynamic nanoscale assemblies of sphingolipids, cholesterol, and proteins. Because this controversy is because of two main factors. First, this was debated because first there was no clear evidence, like in back in the day there was no clear evidence of existence of lipid rafts in vivo. So so though that is because they are very transient and they are usually 10 to 200 nanometers in size, so size also varies on a large scale. So they are, they are nano particles, like they are nano clusters that are present in the plasma membrane. That is why, and again, they're transient. So due to transiency and small in size, they are often below the optical resolution, and due to that there was no clear evidence about the existence of those lipid rafts in vivo. And that is why often these DRMs, they were considered as a controversial topic. But then critic argued, rather than terming them as lipid raft, which lacks the precise biophysical definition, because they do not, as I said, they do not have a clear composition, because they are more about the lipid-lipid and lipid-protein interaction, which makes the membrane rigid, which makes them detergent resistant, rather than having a fixed composition. So when you say lipid raft, it doesn't actually mention a biophysical entity, and that is why they were redefined as the dynamic nanoscale assemblies. Okay, so they were redefined as dynamic nanoscale because they are scaled from 10 to 200 nanometers. So they were termed as dynamic nanoscale assemblies. No, assemblies, because the composition is not clear. So assemblies of sphingolipids, cholesterol, and proteins, right? So scientifically, researchers prefer this term more than lipid rafts, but it is convenient to call them lipid raft. But this is just for your information that due to certain challenges of visualizing those lipid rafts in vivo systems, and arguing about the fact that lipid rafts do not have a fixed composition, because the composition of sphingolipids and phosphatidylcholine, the phospholipids in them, could vary, and they still will have detergent resistant membrane. So for example, let's say that there are you know two lipid rafts with different composition. Let's say in the composition of sphingolipids to saturated phospholipids to cholesterol in one membrane is, let's say, 1:1:2, while in other membrane this composition, saturated fatty acid to cholesterol, this is, let's say, 1:1.5:2. In both of the cases, they are going, the microdomain that is formed by this composition and this composition, that is going to be stable, that is going to be detergent resistant. So they won't get solubilized, and that is why we are terming both of them as lipid rafts. So this often leads to confusion because we cannot define them as a biophysical entity. Whenever you say microtubule, what is the thing that comes to your mind first? That it is a double membrane organelle which which is going to do certain tasks. But when you say lipid rafts which do not have a clear cut fixed composition, so they do lack a precise biophysical definition, and due to this the researchers reterm that as dynamic nanoscale assemblies of sphingolipids, cholesterol, and proteins, right? So this seems more accurate. This was controversy regarding lipid rafts, but besides that, lipid rafts are very, they are very important because they have a lot of signaling molecules and they are important in those downstream signaling cascades. So this is the composition of lipid rafts, and it is important to know about the composition because of all those factors, those proteins, those certain types of lipids, and the signaling molecules. Due to all of them, the rigidity of the membrane is going to increase. That makes this lipid raft resistant to detergents. So now learn about the properties, the biophysical properties of the lipid rafts. So lipid rafts, they are liquid order phase. Okay, so we do not exactly call them fluid, but we call them as liquid ordered phase. So there are different phases of matter, right? Solid, liquid, and gas. So the lipid rafts, they are liquid ordered phase, LO phase, because they are tightly packed, ordered liquid, but they are liquid, right? So it means they are mobile. So even though they are tightly packed, just like solid, so yeah, the order of the packing will be less compared to solid, obviously, because it is a liquid, it is a fluid, but they have, like, unlike the normally lipid bilayer, the plasma membrane, it has lipid bilayer packing, but it is not that much ordered. In the lipid rafts, this order of packing is increased. They are tightly packed, and even though they are tightly packed, they are mobile, and that is why it is called liquid ordered phase. So they are tightly and orderly packed, but still mobile, and hence referred as liquid ordered phase. We already, the size of the lipid rafts is 10 to 20 nanometers. So they are also referred as nanoclusters, the nanoclusters that are present on the plasma membrane. So I also referred that they are transient. So by the transient, I mean the assembly of those lipid rafts is transient. Means let's say that this is the plasma membrane and these are the lipid rafts. So multiple small lipid rafts, they can come together to form a larger microdomain, larger lipid raft, or a larger lipid raft. It can break down to form multiple smaller microdomains, multiple smaller lipid rafts, and that is why I called the lipid rafts as the transient entity, because they are going to form a larger entity or break down into the smaller entity. And that is why the formation or the aggregation of those nanocluster, it is transient, because it is made up of lipids and proteins which are mobile. So they are mobile, they are showing fluidic property, and that is why lipid raft is also transient in nature. So for studying lipid rafts, the techniques that we use is FRAP. We have discussed about FRAP in detail in the previous video, so you can refer to that. Then we can also use single molecule tracking. We can also use atomic force microscopy, or we can use FRET. FRET is fluorescence resonance energy transfer. We haven't yet discussed about FRET. I'll make a video about that. Okay. So those different techniques like FRET, FRAP, atomic force microscopy, and single particle or single molecular tracking. Those different types of techniques could be used. The storm or palm, these are the super resolution techniques. Those different techniques could be used for the visualization of lipid rafts. So now the experimental evidence can also be, it is literally there. We can use Lorden dye, we can use cholesterol depletion, like the way we saw in the liposome formation. So using those different methods and different techniques, we can clearly say that lipid rafts exist even in the in vivo systems, even though it is not it is difficult to visualize them because they are often below the optical resolution. But there is evidence, there is enough experimental evidence to show the formation of lipid rafts. So by this I conclude this lecture. Oh well, we did not discuss the rules about lipid rafts, so let's quickly discuss about roles of lipid rafts. As I said that lipid rafts has signaling molecules. So one obvious function that they're gonna do is signal transduction. So they are going to carry out the downstream signaling process because they have the cluster receptors. They have like the T cell receptor, B cell receptor. They even have a receptor tyrosine kinases. So due to all of them, those cluster receptors, the signaling is efficient. And again, even though there are receptor present, and the lipid rafts, they can serve as platform. The way scaffold proteins serve as a platform for different proteins to go and attach to them, the signaling molecules or the lipid rafts, they can they can serve as platform for kinases, because they can accommodate transmembrane proteins, right? So they can perform as a platform for kinases, for adapter proteins, for which are going to recruit certain other proteins. So that is why they help in signal transduction. Again, lipid rafts, they are also, due to them, there's also pathogen entry. So they help in pathogen entry. They are usually used by viruses such as HIV, influenza. They can be used like the bacteria such as salmonella, or they can use so different types of viruses or different types of bacteria. They use this lipid rafts for the entry, for entering the host cell. Then as I said that there is caveolin and caveolin-like molecules in the lipid rafts. Due to them, there are different types of raft. There is type 1 lipid raft and type 2 lipid raft. Okay, so type 1 lipid raft has caveolins, while type 2 lipid rafts, they have flotillins. So due to the presence of different types of molecules, there are different types of lipid rafts. So the lipid rafts that has caveolin, they mediate the lateral independent endocytosis. So lipid rafts, they also mediate endocytosis and trafficking. So they have a role in trafficking as well, because they mediate endocytosis. So the clathrin dependent, no clathrin independent endocytosis or caveolin dependent endocytosis, that is shown, that is seen in the lipid rafts. So they live, and besides all those factors, beside all those rules, the lipid rafts, they also show, they have also role in immune responses. Okay, so they modulate immune synapse formation, the T cell activation, well, because they have the T cell receptors, and they also, help in antigen presentation. So they are also known to evoke, to have a role in immune responses. So those are the different roles of lipid rafts. So with this, I conclude the lecture. In next lecture, we'll learn about the membrane proteins that are present in the plasma membrane in detail. Till then, see you. Bye bye.